Your blood type is not random. It exists because your ancestors lived in a very specific place, at a very specific time, and faced very specific threats to survival. Blood types didn't appear for identity. They didn't appear for personality. They appeared because human bodies were forced to adapt. If your blood type is O, your family history likely traces back to people who lived in a world of constant movement. Migration, hunting, injury, and sudden death. Survival depended on reacting fast and bleeding less from internal damage. If your blood type is A, your ancestors were more likely part of humanity's first permanent settlements. Farming brought food security, but also crowding, waste, livestock, and slow-spreading disease. Survival now depended on immune systems that could tolerate long-term exposure. If your blood type is B, your blood carries genetic markers linked to migration across vast distances, Central Asia, steppe routes, and harsh, changing environments where adaptation mattered more than stability. And if your blood type is AB, your family history only exists because very different populations eventually met, mixed, and reproduced. This is the newest blood type and the rarest evidence of human movement and blending late in history. This is not speculation. These patterns come from population genetics, ancient DNA analysis, archaeology, and modern medical research. Scientists can map blood type frequencies across continents. They can extract DNA from ancient skeletons. They can compare immune responses, disease risk, and survival patterns today. And when they do, the same picture appears again and again. Blood types follow migration. They follow settlement. They follow isolation. They follow mixing. Your blood type is not about who you are today. It is about what your ancestors had to survive long enough to reproduce. In this video, we're going to trace what your blood type reveals about your family's past. Not through myths, not through personality theories, but through evidence. And to understand that story, we have to go back to a time before blood types even existed, when humanity was still one small population, facing the same dangers and bleeding the same way. A world where survival had no backups. For most of human history, there was no safety. No walls, no farms, no stored food, only movement. For more than 200,000 years, humans lived as hunter-gatherers. This is confirmed by archaeological evidence and ancient DNA research across Africa, the Middle East, and Eurasia. Small groups moved constantly. They followed animals. They followed the water. They followed seasons. And they faced danger every day. Injuries were common. Hunting accidents, animal attacks, falls, open wounds. There were no antibiotics, no surgery, no hospitals. If bleeding didn't stop, you died. If infection spreads, you die. If parasites took over, you died. In this world, survival depended on reacting fast. This is where type O blood dominated. Genetic research on the ABO gene, published in Nature, Genetics, and PNAS, shows that type O is the oldest and original human blood type. It existed before agriculture, before villages, before civilization. Type O blood lacks both A and B antigens. That absence changes how the immune system behaves. According to studies from Harvard School of Public Health and Infectious Disease Research published by the World Health Organization, type O blood is linked to stronger early immune responses. Not subtle, not controlled, aggressive. This mattered in a world full of parasites and sudden infection. Malaria research offers one of the clearest examples. Large population studies in Africa show that type O blood provides partial protection against severe malaria. This finding has been replicated across multiple studies and is widely accepted in evolutionary medicine. In early human environments, malaria was deadly. Any advantage meant survival. Type O blood also behaves differently when it comes to clotting. Modern medical studies show that type O individuals have a higher risk of severe bleeding during surgery or trauma. But in the ancient world, this was not a disadvantage. Why? Because people weren't living long enough for long-term complications to matter. Evolution does not reward longevity. It rewards reproduction. Type O blood helped humans survive injuries, infections, and parasites long enough to have children. As humans migrated out of Africa, this blood type spread with them. Genetic mapping of blood type frequencies shows that type O remains most common in populations shaped by long-term migration and limited early farming. Indigenous groups. Nomadic populations. Regions where settlement came late or not at all. The blood followed movement. Type O ancestry is linked to people who lived fast, moved often, faced constant physical danger, survived through immune aggression and resilience. But this survival style had a cost. When humans stopped moving, life became safer in one way, and more dangerous in another. For most of human history, survival meant motion. If food ran out, people moved. If disease appeared, they left. If the land failed, they followed animals somewhere else. Then something changed. Around 10,000 to 12,000 years ago, in regions like the Fertile Crescent, early Europe, and East Asia, humans made a decision that reshaped biology itself. They stayed. Archaeologists have documented this shift clearly. Excavations show permanent homes, storage pits, farming tools, and animal pens appearing almost at the same time across different parts of the world. This was the agricultural revolution. Instead of hunting wild animals, people planted grains like wheat, barley, and rice. Instead of temporary camps, they built houses meant to last generations. Instead of small bands, they formed villages, then towns. For the first time, humans lived close together every single day, and that changed disease forever. According to research published in PNAS's and Nature Genetics, early farming settlements created the ideal environment for illnesses that humans had never faced before. Waste didn't disappear when people stayed in one place. Animals slept near humans. Water sources were reused again and again. This allowed bacteria and viruses to circulate constantly. Diseases like tuberculosis, early forms of measles, and smallpox did not need to kill quickly anymore. They survived by lingering. Instead of sudden death from injury, people became sick slowly, repeatedly, and for long periods of time. This new world created a problem for the old immune system. Anthropologists and evolutionary biologists agree on this point. Early humans evolved immune systems designed for short, violent threats. A deep cut, a fast infection, a sudden wound. Those immune responses were aggressive. They attacked hard and fast. But in a village, constant aggression became dangerous. A strong immune response causes inflammation. Inflammation helps in emergencies, but damages the body when it never shuts off. Modern immunology research shows that chronic inflammation damages organs, blood vessels, and tissue over time. In farming societies, people weren't dying from sudden attacks anymore. They were dying from long-term immune stress. This is where type A blood begins to matter. Scientists studying the ABO gene, the gene that determines blood type, have found that the A antigen changes how immune cells interact with invading pathogens. Research from Harvard Medical School and the University of Tokyo shows that type A blood is associated with more regulated immune signaling. Not weaker, just less explosive. The immune system still responds, but with more control. That mattered in crowded settlements. People with type A blood were less likely to suffer extreme inflammatory damage from constant exposure to disease. This didn't make them healthier forever. It made them survive long enough. Ancient DNA supports this pattern. Genetic analysis of skeletal remains from early European farming communities shows significantly higher frequencies of type A blood compared to nearby hunter-gatherer groups who continued to move seasonally. The difference wasn't cultural. It was biological. The blood followed the lifestyle. As farming spread, type A blood spread with it, across Europe into parts of East Asia, and throughout regions where people stayed rooted to land. But adaptation always comes with a cost. Modern medical research consistently shows that type A blood is linked to higher risks of blood clots, heart disease, and stroke. The American Heart Association and multiple large-scale population studies have confirmed this association. Why? Because the same biological traits that reduce inflammation can increase clot formation over time. In today's world, where people live long lives, that matters. In the ancient world, it didn't. Most people never reached old age. What mattered was surviving childhood illnesses, infections, and repeated exposure long enough to reproduce. Type A blood did that job. It allowed families to grow roots. This blood tells a very specific story. These ancestors stayed in one place. They worked the same land year after year. They raised children near their parents. They depended on cooperation, shared rules, and shared labor. Villages required patience. Farming required planning. Living close together required tolerance. Civilization did not begin with strength. It began with endurance. It began with bodies that could tolerate constant exposure instead of reacting violently to every threat. If your blood type is A, your family history likely comes from the world's first settlers. People who traded speed for stability, movement for memory, survival for structure. But not everyone accepted this settled life. When humans began farming, not everyone followed. The rise of agriculture created opportunity, but it also created limits. Crops depended on rainfall. Farms failed in cold climates. Grains could not survive high altitudes or dry grasslands. So while some groups stayed and built villages, others moved away from their early farming centers altogether. Anthropologists and archaeologists have traced these movements across Central Asia, the Eurasian steppe, parts of South Asia, and regions bordering the Middle East. These lands were not ideal for crops, but they were ideal for animals. Here, survival depended on cattle, sheep, goats, yaks, and horses. People followed herds. They moved seasonally. They lived in temporary settlements. They did not abandon mobility, but they did not roam randomly either. This lifestyle is known as pastoralism, and it created a very different biological pressure. Pastoral societies lived in constant contact with animals. They drank milk. They consumed fermented dairy. They handled animals daily. They were exposed to animal bacteria continuously. According to research published in Nature, the Journal of Infectious Diseases, and PNS, this kind of exposure changed the immune environment completely. The immune system could not afford to react aggressively all the time. A constant high alert immune response causes chronic inflammation, and chronic inflammation damages tissue, joints, organs, and blood vessels. In these environments, survival depended on immune balance, not immune force. This is where type B blood enters human history. Geneticists studying the ABO gene have identified a mutation that altered the structure of red blood cells in these populations. This mutation added a new antigen. That antigen became known as type B. Type B blood changes how immune cells recognize and respond to certain bacteria, especially those associated with animals and fermented foods. Studies from immunology departments at institutions like University College London and the Max Planck Institute show that type B blood is associated with more selective immune activation. Not weaker, not stronger, more targeted. This mattered enormously in pastoral societies. People needed to tolerate constant low-level exposure to microbes without triggering destructive inflammation. Over generations, this mutation spread, but only where the lifestyle supported it. Type B blood did not spread evenly across the world. It remained uncommon in early farming centers like Western Europe. It stayed rare in populations that relied heavily on grains, but it became far more common in regions tied to herding and seasonal movement. Today, the highest frequencies of type B blood are found in Central Asia, parts of South Asia, and areas connected historically by migration corridors and trade routes. The distribution is not random. It follows ancient movement. The blood reflects adaptation, not stability. Type B ancestry is linked to people who did not fully settle and did not constantly flee. They adjusted. Their survival depended on flexibility, on switching food sources, on tolerating new bacteria, on surviving changing climates year after year. This blood type did not arise from comfort. It arose from balance. Modern medicine still sees traces of this history. Type B individuals show different metabolic responses to certain foods. Some studies suggest different immune reactions to specific infections. These are not personality traits. They are biological echoes of an adaptive past. And eventually, history did something it had rarely done before. Populations that had stayed, and populations that had adapted, finally met. Trade expanded, migration increased, roots crossed, and when people with type A blood and people with type B blood began having children together, a new blood type appeared, not through slow evolution, not through thousands of years of isolation, but instantly. A blood type that carries both histories at once. and that blood type would tell the most recent story of all. Type AB blood did not evolve the way the others did. It did not appear because humans entered a new climate. It did not rise because of a new food source. It did not spread because it offered a survival advantage. Type AB exists for one reason only: human mixing. From a genetic standpoint, type AB blood is the simplest blood type to explain, and the hardest to create historically. The ABO system works through inheritance. If one parent carries type A blood, they pass down the A antigen. If the other carries type B blood, they pass down the B antigen. When a child inherits both, the result is type AB. No mutation is required. No slow adaptation. No environmental pressure. Just contact. And that detail matters. Because for most of human history, type A and type B populations did not meet. Early farming societies were localized. Pastoral groups moved, but within defined routes. Mountains, deserts, oceans, and climate kept populations apart for thousands of years. Anthropologists have shown that even populations living a few hundred miles apart could remain genetically isolated for centuries. So, while type A spread through early agricultural zones, and type B spread through migratory and pastoral regions, there was very little overlap. That began to change around 4,000 to 5,000 years ago. Archaeological evidence shows a sharp increase in long-distance movement during this period. Trade routes expanded. Metal tools improved transportation. Domesticated horses increased mobility. Early empires began pulling distant populations together. Ancient DNA studies from the Max Planck Institute for Evolutionary Anthropology and Harvard's Ancient DNA Project show a clear rise in genetic mixing across Europe, Central Asia, and the Middle East during this time. Skeletons from this era contain genetic markers that simply did not appear together before. This is when type AB begins to appear in the record. And this explains why type AB remains rare. Even today, type AB is the least common major blood type worldwide. In many populations, it exists in less than 4% of people. That rarity is not accidental. It reflects how late and limited large-scale human mixing was for most of history. From a biological perspective, type AB is unusual. It carries both A and B antigens, which means the immune system must tolerate a broader range of markers on red blood cells. This affects how the body responds to threats. Studies published in the New England Journal of Medicine, the American Journal of Hematology, and large population health analyses show that type AB blood is associated with altered inflammatory signaling, higher baseline risk of certain clotting disorders, distinct immune responses compared to type A or type B alone. But none of this suggests optimization. Type AB was not designed to be better. It was created by connection, by people crossing boundaries that had existed for thousands of years. Medically, this blood type reveals its mixed nature clearly. People with type AB can receive blood from both type A and type B donors, but they can donate only to other AB individuals. A wide intake, a narrow output. Even in modern medicine, this blood reflects integration without dominance. Type AB family history tells a very specific story. Your ancestors came from different biological worlds. Some likely descended from settled farming societies, others from migratory or pastoral populations. They ate differently. They faced different diseases. They lived under different survival rules. And at some point in relatively recent history, those worlds met. Not through conquest alone, not through slow drift, but through trade, migration, marriage, and shared space. Type AB blood is evidence of humanity entering a new phase, one where separation gave way to connection. It is not ancient. It is not primal. It is modern. It exists because humans stopped staying apart. And in that sense, type AB blood is not just about ancestry. It is proof of a moment in history when human family lines finally began to overlap and stay overlapped. But that does not mean blood types control us. Your blood type is not a personality test. It does not determine intelligence. It does not predict behavior. It does not define who you are. But it does record what your ancestors had to survive. Modern medicine treats blood types as clinical tools. They guide transfusions. They guide organ matching. They guide pregnancy care. But beneath those medical uses is a deeper pattern, one that only becomes visible when millions of people are studied together. Large-scale population research shows that blood types still influence how the human body responds to disease, injury, and long-term stress. Not in isolation, not as fate, but as probability. For example, multiple studies published in The Lancet, Circulation, and Nature Medicine show that people with type O blood have a lower risk of heart disease and stroke, but a higher risk of severe bleeding after trauma. This matches what evolutionary biologists would expect from a blood type shaped in a world of injury and sudden danger. Type O blood flows freely. That protected the heart, but it made stopping bleeding harder. Type A blood shows a different pattern. Research consistently links type A to a higher risk of blood clotting, cardiovascular disease, and inflammatory conditions. This reflects a blood type shaped not by sudden trauma, but by long-term exposure to disease in settled populations. Type B blood shows yet another pattern. Studies suggest different immune responses to specific infections and differences in how the body handles inflammation and metabolism. This fits with a history shaped by movement, animal exposure, and environmental change. And type AB, the rarest and youngest, shows unique immune and cognitive risk profiles in modern studies, including higher rates of certain inflammatory and neurological conditions. Again, this is not destiny. It is residue. Modern doctors do not treat patients based on blood type alone. But blood type is never ignored, because it still carries information, and this is where family history becomes visible in the present. When geneticists map blood type frequencies across continents, they see clear patterns. Type O follows early human migration routes. Type A clusters around ancient farming zones. Type B tracks pastoral corridors and trade paths. Type AB appears where populations mixed late in history. These patterns match archaeological records. They match ancient DNA. They match known human movement. Blood types act like biological fossils. They preserve traces of events that written history never recorded. Ice age crossings, agricultural expansions, nomadic migrations, empires rising and collapsing, people meeting who had never met before. Your blood does not tell the story of you. It tells the story of before you. People who lived without antibiotics, without anesthesia, without modern medicine. People whose survival depended on how their bodies responded to injury, infection, and environment. Most of them never knew why they lived or died, but their biology learned. And that learning was passed forward, not as memory, not as culture, but as structure. Your blood type is not an advantage. It is not a weakness. It is evidence. Evidence that human history is not only written in stone and paper, but carried quietly inside living bodies. Your blood is not random. It is a record. And when you understand that, you are no longer just looking at veins and cells. You are looking at human history, still flowing,